Welcome back, children. Last video we have learned about microbes in household products. In this video we are going to learn about microbes in industrial products. Okay, what are the three industrial products we are going to see? Means we are going to learn about fermented beverages and antibiotics, some of the enzymes, chemicals, and also some bioactive molecules. So I'll cover all those microbes which can be used in industries only in this video. So one video even I'll cover microbes in industrial products. So let's get into class. Okay, here all those microorganisms what we have learned for household only, no, in that, and also some different kinds of microorganisms will be involved in industrial products. Okay, now look at the slide. First point, production on an industrial scale requires growing microbes in a very large vessels called fermenters. So what we used to do in our household or lab, that is all a small scale. We'll use fermenters or we'll use small vessels for our purpose, but it is not in the case of industries. We used to have a large fermenter. Okay, large vessels only we can call it as fermenter. So for fermentation process we are using large vessels. These kinds of large vessels we can call it as fermenters. Is that clear, children? Now I will explain the point. Now look at this. This is the fermenter. So these are the large vessels which can be used in industries for the production of our desired product. First we will see fermented beverages. Okay, if I tell fermented beverages means automatically for everyone's mind it will come only one fungi yeast. Okay, only one fungi yeast. It is a single cellular. We all know, no? It is a single cellular. And what is the scientific name for that? Saccharomyces cerevisiae. Okay, what is the scientific name for yeast? Saccharomyces cerevisiae. And it is used for what and all? Bread making, and also brewers yeast. That is only it is available in so many markets. We can also get and we can use it for our fermentation process, and it will be used for industrial purpose also. And it is used for fermenting malted cereals. So what do you mean by malted cereals, children? So here we used to soak cereals for for 8 hours. Afterwards it started to form sprouts. So that sprout forming cereals, okay, formation of sprouts from cereals only we can call it as malted cereals. And fruit juices to produce ethanol. So we are using either malted cereals or fruit juices to get ethanol with the help of a fungi called Saccharomyces cerevisiae. So this process is called fermentation. This can be done largely in industries through a fermenter. So here material is what? Either malted cereals. Malted cereals might be anything. We can use ragi, we can use whatever cereals what we want. And we are using the baker's yeast, that is brewer's yeast, Saccharomyces cerevisiae, that is a fungi. So here what is the starter material, children? What rich material we are taking? We are taking a carbohydrate rich, like, and also the fruit juices we are taking. So from these they all contain a carbohydrate containing compounds. From that we easily will get C2H5 compound, C2H5OH. So we'll get alcohol product. So we'll get ethanol. And here first you want to remember malted cereals or fruit juices to get ethanol with the help of brewer's yeast or baker's yeast. And here second product is wine. Okay, wine and beer are produced without distillation. Wine or beer is produced without distillation, whereas whiskey, brandy and rum are produced by distillation of the fermented broth. Here you remember. So now we are taking fruit juices or we are taking malted cereals, whatever the process. After a few days, after a 15 days, we will get a one broth. So imagine this is the broth. Now look at this conical flask. This is a broth. Broth means a medium. Okay, medium. So after a fermentation, we'll get our product now. So that product only we can call it as a fermented broth. So this fermented broth either we can use in two ways. Using distillation, using non-distillation method. So if we are filtering the fermented medium, if we are filtering the fermentation product, fermented product as such, and when we are consuming means that we can call it as wine and beer. So here there is no distillation. We won't go for any distillation process. After fermentation, we are filtering the nutrient rich liquid, okay, nutrient rich liquid, and also fermented product, okay, everything we are filtering. So the filtering, as such filtering we are using means that we can call it as wine and beer. We are not going for any distillation process. And second category, whiskey, brandy and rum and all, will go for distillation process, where only alcohol alone will be evaporated, and again will condensed. We will get only alcohol rich product alone. That is only we'll call it as whiskey, brandy and rum. So whiskey, brandy and rum are, because of distillation, will get only alcohol rich alone. It undergone condensation process, first evaporation and then condensation, will get only the ethanol alone, means that is distillation, whiskey, brandy, rum. And wine and beer, no distillation, so we'll get we won't get only alcohol, along with some nutrient will be there, okay. That is only we'll call it as wine and beer. Is that clear, children? So we have learned about the yeast role, and also what is fermentation process, and fermentation process will get alcohol, ethanol, or we can tell alcohol. So we'll get in two form, through distillation and without distillation. With distillation means that is your whiskey, brandy, rum. Here evaporation, condensation, so only alcohol, C2H5OH alone you will get. But it is not in the case of wine and beer, where we can get along with alcohol some nutrient also. Is that clear, children? So next we'll move on to the next slide. So that is about your fermented beverages. Is that clear? So under microbes in industries, now we have learned about fermented beverages. Example for fermented beverages is your whiskey, brandy, rum, and also your beer and wine. And what is distillation? What is without distillation? How we got the other like beer and all? Only their filtration alone. Next we'll move on to the antibiotics. So already we have learned in grade 8 also. So what is antibiotics? Anti means against, biotic means living things. So against living things, what we are giving medicine, that is only we can call it as antibiotics. Who first discovered antibiotic? Alexander Fleming, okay, and he only discovered antibiotic. So what is the name of the antibiotic? Penicillin. So from which fungi he has discovered? Penicillium notatum. Penicillium notatum, in that fungi only he has discovered a first antibiotic. And we can take penicillin from Penicillium chrysogenum, that is another species. So Penicillium notatum is a common species, or we can take Penicillium chrysogenum. From that also we can take penicillin from the fungi group. So that is the first antibiotic, and it is not an discovery, it is an accidental discovery only, okay. So it is an accidental discovery. So we are going to learn about the antibiotics in detail. Now look at this Petri plate, children. So this Petri plate, this Alexander Fleming, he has cultured some bacteria. But he didn't wash that Petri plate properly, and some of the bacteria was left as such. And this penicillin, it started to eat the bacteria which is present in the Petri plate. In that only he came to know this fungi, mold, okay, this mold can able to kill some of the bacteria. So penicillin is a fungi antibiotic, can able to kill some of the bacterial diseases caused by streptococcus, okay, streptococcus, staphylococcus bacteria, like that. Because of bacteria, what kind of disease we get? It was killed by the penicillin antibiotic. So this is a Petri plate. Now penicillin colony, now look at that Petri plate. In that penicillin colony there is no bacteria at all. So from that only he came to know the penicillin act as an antibiotic to kill the bacteria. So it is an accidental discovery. So antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease causing microbes. So either it will kill or retard. Retard means it will stop, it will reduce, okay, kill or retard the growth of other disease causing microbes. And penicillin was the first antibiotic to be discovered, and it was chance of discovery. That's why we can call it as accidental discovery. And Alexander Fleming, while working on Staphylococci bacteria, okay, once observed a mold is fungi growing on one of his unwashed culture plates, around which a Staphylococci could not grow. He found out that it was due to a chemical produced by the mold, and the name of the chemical is penicillin. So from penicillium a chemical is produced, that is penicillin. Later, after the mold, Penicillium notatum. So later, first he discovered by chance. Afterwards, two scientists, okay, Ernest Chain and Howard Florey. Ernest Chain and Howard Florey, they made it a full potential effective antibiotic. So first it was discovered by accidentally. Afterwards, it was 100%. It was achieved. It was done by the Ernest Chain and Howard Florey. For this work, both Fleming and also the two scientists, Ernest Chain and Howard Florey, they were awarded Nobel Prize together. Okay, children. So that is only penicillin. Now I think this is the structure of penicillin. So next we'll move on to the. So this it was discovered in the World War time. So it was much useful for the soldiers who and all affected by the disease. That's why the picture was given. Thanks to penicillin, he will come home. So because of that, so many people, they got their life. Okay, next we'll move on to the chemicals, enzymes and also other bioactive molecules. So here I have told penicillin, it will kill bacteria, means it will kill most of the bacterial diseases like plague. Some of the example was given in your book. It will kill plague and also some whooping cough, diphtheria, cholera and also leprosy. Okay, it was all those days, so many people, they were affected by these kind of diseases, and they all get rid of because of the wonderful discovery antibiotic. So during World War II it was happened. Okay, children. So next we'll move on to the chemicals, enzymes and other bioactive molecules. So here nothing to explain, but you want to remember so many names alone. It will all be interesting. So here first we will see Aspergillus niger. It is a fungi. It will help to form citric acid. Normally we used to take citric acid from lemon only, but industrial they will produce organic acid. So if I tell all those acid, citric acid, acetic acid, butyric acid, lactic acid, all these acids, not this one, all these acids are, we can call it as organic acid. Why is it so, children? Because why we can call it as organic acid? Because we are getting all those conversion based on the carbohydrate rich component. So we are getting from the organic component. So all those acids we can call it as organic acids. So what is the first acid? Citric acid. We are getting from one fungi group, that is Aspergillus niger. In industry, normally from fruit, from vitamin C fruit, we'll get citric acid. Like lemon, orange, everything. But in industrial also we need a citric acid, because citric acid acts as a good preservative. So for preservation we need citric acid. For that industrially they are using Aspergillus niger. And Acetobacter aceti, I have told, acetic acid, Acetobacter aceti, acetic acid. And Clostridium botulicum, it will help to form a butyric acid. Clostridium botulicum is help to form a butyric acid. And Lactobacillus, that we are familiar, lactic acid. So 4 acid also organic acid, and normally these 4 are bacterium. And Saccharomyces cerevisiae, it is a fungi, is helping to get ethanol, that is your fermented product, alcohol. Okay, this is the some of the chemicals what we are getting through the because of your microbes. So these are the some chemical example. Is that clear, children? Chemicals. Okay, next we'll move on to the enzymes. So we all know what is enzymes. Enzyme is helping, enzymes of proteinaceous nature, is helping to do catalyst so many reactions and help to break cell wall, break to cleave cell wall. So many function is behind enzymes. That is all our familiar examples. Apart from that, it is used in other areas also. Now imagine lipase. Lipase is an enzyme. It is used in laundry detergents. Okay, so it helps to remove so much of dirt which is present in your clothes. So it can be used in a laundry detergents. And pectinase and proteases, it is used in bottle juice. Now look at so many we will get from Sharpno orange juice and apple juice, strawberry. It is all very plain, children. Okay, no cell debris will be there. Why? How we are getting all plain, neat juices means because they'll use pectinase and proteases. So these kind of pectinases and proteinase enzymes, no, it will break the cell debris which is present in the juices. So we'll get a neat plain juices. So pectinases and proteases, because we are taking from fruit pulp only, no. So fruit pulp is made up of a pectinase, a fruit cell one made up of pectinases, and some of the protein will be present over there. So all was broken by the pectinase enzyme and protease enzymes, and we will get a bottled juice. So, but when we compare to our normal orange juice, no, our orange juice is different from the bottle juice. Why? We are not using any enzymes to break all those components. As such, we will consume. Our body will take care of all those things. But come to market means they need a very neat and filtered juices, no? For that they are going, because filtered also, and you need some little bit solid also. If it is very watery means automatically we won't get taste. So it needs some semi-solid, like little solid and watery. So how they are achieving means they'll add a pectinase enzyme, protease enzyme to cleave, and they'll get a product. So that is the principle they are using for bottle juices. And streptokinase, okay, that is an enzyme they are getting from a streptococcus bacterium. And here this very very important, children, this streptokinase enzyme, it is used as a clot burster to remove clots in your the blood vessels. Now this, no, it will burst, it will help to burst the clot. Adipella irundha, it will automatically, it will help to remove. So the streptokinase is an enzyme which can be taken from the microbe streptococcus bacterium. So lipase, pectinase, protease, streptokinase, all those enzymes were taken from the microbes for our different purposes. Normally lipase, no, it used to remove your oily stains, okay, because without knowing your costly dress and all affected by oily stains means it will be removed by lipase enzyme. And also our fruit juice is different from our bottle juice because they have used pectinase and protease enzyme. And a very important clot buster is your streptokinase enzyme, which we are taking from streptococcus. Okay, and it is very important to prevent a person from a heart attack. If not, a person will be affected by heart attack. So next, last part, bioactive molecules. So what do you mean by bioactive molecules? So here some molecules will be produced, and it is very very important in the clinical studies. So here one such bioactive molecule is cyclosporine. Okay, cyclosporine A. It is a bioactive molecule. It is not a chemical, it is not an enzyme, it is a molecule only. Cyclosporine A. Okay, and what is the role? Where we are taking means we are taking from the fungi group. Okay, Trichoderma polysporum. Trichoderma polysporum is a fungi. From that only we are taking cyclosporine A, a bioactive molecule. And what is the role? Here the role is immunosuppressive agent. So what do you mean by immunosuppressive agent for which people? For organ transplant patients. What do you mean by immunosuppressive agent means? Immunosuppressive agent means we are for a short time we are suppressing our immunity power. Because when we go for organ transplantation, automatically some new organ, okay, some new material is they are going to introduce into your body, a new organ. So it is a foreign particle for your body. So if once a foreign particles were introduced into your body, automatically your body started to produce antibodies against the antigens. Okay, similarly organ, because this organ might be their relative or friends or unknown persons, they might contain some of the antigen particle also, and whenever it is added, automatically your body started to produce immunity, some reactions in your body. So that is all unwanted reactions only. So we need to suppress our immunity power by giving some immunosuppressive agent. What is the immunosuppressive agent here? Here a agent is cyclosporine A. I'll explain one more immunosuppressant agent also. For example, if a person is exposed to coronavirus means automatically the person body started to synthesize so many cytokine enzyme. Cytokine protein, not enzyme, cytokine protein. So the cytokine protein, what it will do means it started to clot the blood. Why? Because more in number in the cytokine will reduce the RBCs, will reduce the count of other cells. And this cytokine, and if there is no movement of blood, means all your organs started to failure. So within a 4 minutes, organs lose its viability, and automatically the person, they can't able to, if suppose blood is not going to lungs, means a person can't able to breathe normally. So that's in that condition, like the person will be affected by suffocation. So this so we need to suppress the immunity power of our body. Okay, while suppressing only we can get rid of from this problem. For that, doctors used to prescribe, not subscribe, used to prescribe a medicine called immunosuppressive medicine. So this immunosuppressive medicine will stop the synthesis of cytokine protein in your body against antigen, because if too much is also bad for your health, children. Antibodies means only they should produce in a proper level. In that condition only they can able to fight, because it is in the blood. They should travel throughout our body. If more antibodies in your blood is also, it's a problem only, because it will not allow to move the red blood cells to the organs, and the red blood cells only carrying oxygen. So it is all become problematic. So we even antibodies also should be produced in a normal rate only. When it is cross only, we'll get a problem. And early detection can be done for the appearance of cytokine in your body, and for that only doctor used to prescribe the immunosuppressive agent. So what you want to remember here means, immunosuppressive agent means we are suppressing our immunity power by introducing like a cyclosporin A, we are getting from the microbe. So microbe is very, very important. And second one is statin. Statin is a monococcus purpurus yeast. From that we are getting statin, and it is also again very important biomolecule, helping to reduce the cholesterol in your blood. Okay, cholesterol in your blood. Is that clear, children? So that's all about the microbes in industry. So next video I'll send microbes in sewage treatment. With that we'll stop. So afternoon I can send the other two. Thank you, children.